the simplest protocol is hybridization, differential hybridization of a probe to a sample. So here you can see a sample that's got an A on it and a sample that's got a G, just like in the previous example. If you have a probe that comes in that's got a T on it, it'll bind here, and it will not bind nicely here, as shown by the fact that there's a bulge in the DNA. Whereas you can see this is the second probe, which has got a G. It binds to a probe that's got a C there, whereas the A sample does not bind with a C. And what makes it, what allows you to distinguish these two reactions occurring, the two alleles in this case, is that this allele is labeled, this probe is labeled with one color, and this is labeled with a second color. These are fluorophores that in a lab you can detect with filters that measure one color versus another color of light. In the reaction, these probes, if they bind perfectly, are cleaved. And the last thing I need to tell you is that in this probe, you have a fluorescent molecule and also a quencher molecule. This quencher molecule interacts with that fluorescent molecule and keeps it from generating color. But during PCR, this probe, which sits down on the amplicon that's being made, gets cut if it's perfectly matched. And the fluor and quencher are separated and light is generated. And I won't go through the other technologies because they're really just variants of the same thing. The bottom line is that if you've got an A sample, you generate this colored light. If you've got the G sample, you generate the other colored light. And if you have a heterozygote, you would generate both colors of light. So now you've taken the fact that this hybridization event depends upon sequence, and you've turned it into a color you can see. Now, unfortunately, this was supposed to be a figure which showed you actual data. I wasn't going to tell what the gene was because it's secret, but it's data that I've generated for Esteban as part of his Gala study he's going to tell you about, and it shows a nice distribution for 384 samples. But to give you an idea of like what this is like in the lab, essentially, just picture large stacks of plastic plates that have got 384 little wells in them, and robotics to be able to transfer and mix and heat and read these samples. It's actually quite prosaic in a way, although doing it well can be a thing of beauty. At least I think so. And the technology itself, I tell people who are thinking of getting into it, no scientist in the lab who does this has any more hands than you do. It's not difficult. However, that doesn't mean that it's necessarily trivial. I think the important thing here is kind of careful control, careful validation, and so on. That was an instance of a technology you can use to look at lots of samples times one SNP. But what if you want to look at thousands of SNPs? And you run into a problem there. To look at thousands of SNPs would take you months or years using a technology such as I just showed, which is very flexible. You can rapidly look at some new gene, and that's the kind of stuff that Esteban's been doing. But in the future, he's talking about looking at thousands of genes at the same time. And technologies like the TACMen assay, which is the name of the assay I just described, do not scale appropriately for looking at thousands of SNPs. And of course, that's where we're going. For that, of course, you can take a technology, and I'm not going to go into the details here except to say, here's a detection technology. In this case, I think it's the mini sequencing approach that results in different colors. And you can, on a microscope slide, have different spots that have got different capture probes that will separate and localize these differently labeled molecules to different spots on the slide. And this is shown here. Different spots on the slide can be queried by a laser, for instance, and you can get information about that, about thousands of samples at the same time. There are a lot of different ways these arrays can be made. This is one example of a technology that I've recently been playing around with and that Esteban plans to use, where literally on a microscope slide they etch tiny little holes using an acid-based etching protocol, and then they add glass beads, just like a Chinese checkerboard. And each glass bead has been synthesized separately to have a specific capture molecule on it that queries a specific single assay. When you add those beads to this Chinese checkerboard-like surface, they all fall into the holes and stay there, and that allows you with a laser later to read each one of these beads, and each one represents a different assay that are all done in one big multiplex, as they're called. So this is the kind of technology that allows you to go to the next level of analysis. OK, this is just to show you that I've just shown you two technologies. If you want to look at it in depth, there are a huge number of different reaction principles, formats, detection methods, companies. It's bewildering. And this is very much, I think, like any technology in the early stages. When there's not been a lot of standardization and a lot of competition, there's a lot of ferment, and it's quite exciting. And I think that that paper does give you more detail about the different technologies. It doesn't really matter. I wouldn't spend a whole lot of time trying to learn these. It just goes to show that there are a lot of different approaches and that none is perfect. This, for instance, is an example of the kind of thought process that went into the selection I've made, because of course you can't afford to have all of these systems at once. This shows you that for low numbers of samples versus high numbers of samples, and low numbers of SNPs versus high numbers of SNPs, you might want to use different kinds of technologies to cover all the different kinds of study. And of course, all of these kinds of study are valid and important. So for instance, I've got these, the TACMAN method I told you about and the Illumina method, and you try to cover the space as best as you can. OK, now let me just briefly go over microarray expression. That was genotyping, and that's the way that you can look at differences in the sequence that exists between people. The other kind of analysis in functional genomics is gene expression, where you might want to ask, of 30,000 genes, which are expressed in a certain tissue? And the way that that's done is also generally using microarrays. And I'm not going to go over this except to say that microarrays, like the arrays I just described to you for genotyping, are flat substrates in which you've made lots of little spots. And there are a lot of different ways to make them, as I kind of showed in that diagrammatic figure. And each one has got a different sequence that allows you to measure the amount of the transcript, the mRNA of a given gene. And that gives you data, ultimately. And typically, this data can be for 1 or 2 samples that are analyzed at the same time. So it's kind of much slower. It's very difficult to look at thousands of samples by microarrays. But you can look at thousands of genes at the same time quite easily. So it's a slightly different format than genotyping. And of course, it's for a completely different kind of question. How am I doing for time, by the way? OK. I think I might skip a little bit of this primary data, which is on microarray gene expression. I'm just going to show you this figure. This is the type of data output you get from a traditional microarray experiment. This is a log 10 scale of abundance of a transcript. In other words, you're talking about a span of 4 orders of magnitude of mRNA concentration from one sample and from another sample. In this particular experiment, it was a knockout mouse that's missing a key antioxidant enzyme. Sorry, this is the wild type, and a knockout. And these animals have got a very dramatic phenotype. They die actually about a week after birth. And the question was, well, which genes are expressed differently in these mice? Essentially, if there was no difference between these 2 mice, you'd expect that each of these data points represents one gene. And this would be the expression level in the knockout mouse versus the wild type versus the knockout. And if there was no difference between the 2, this would be a very, very tight line that went right up the middle of the plot, because it would mean that there was no difference between the 2. To the extent that there are genes that vary on either side of this, that means that they're expressed at a higher level in the wild type than in the mutant, or at a higher level in the mutant than in the wild type. A couple take-home messages here. First of all, you can't look at this kind of information in a traditional way. I mean, you can't print out a list, for instance, of 30,000 genes and just kind of look through it. Also, you have a statistical problem that, and this is extremely valid for gene expression and genotyping. You get into a problem of multiple testing, where there's a certain degree of variation in running the assay that's always there. And if you're looking at 30,000 genes and you apply a traditional statistical test that if a p-value, for instance, of less than 0.05, then you're going to end up getting hundreds to thousands of false positives. Genes that you think, gee, that's a really differently expressed gene. I should go look at that. And it's merely there because you've looked at 30,000 genes. In fact, with a p-value of 0.05, you'd expect something like 1,000 just by chance. So the statistical algorithms that you need to apply are different. And the data slides that I'm not going to show, and I'll just kind of go through it, have to do with the fact that in the case particularly of gene expression data, the quality of the data themselves is often questionable. And so this is some work that I had done actually when I was working at a company called Gorilla Genomics to actually ask how good are the data. And I'll just skip through it. The bottom line is, and this is another picture that's slightly off, this did show number of samples versus number of genes. And just like with genotyping, you have the same kind of trade-off where there's more than one technology, and different technologies are appropriate for different scales of analyses. Microarrays are great if you have a large number of genes, but they're not great if you have a large number of samples. However, once you know the genes that you're interested in, you can easily look at a large number of samples for a small number of genes by applying another technology called quantitative PCR, which is kind of my specialty.